All right, I'm going to go ahead and share my screen. My name is Jason Biggs. As Jamie said, I'm a developer for Wolfram Alpha and the Wolfram Language, focusing on chemistry content. All right, so I'm going to talk about the ever-expanding Wolfram Knowledgebase, and specifically what I have to do with it, which is Wolfram chemistry. I always like to throw up some kind of random picture of molecules at the beginning of a talk here. Right, so the Wolfram Knowledgebase. In case you were not aware, it's got curated information on over 400 different entity types. Lots of people like to think of them as the little yellow boxes, right? We previously had chemical data, and now we have chemical entities. Well, we don't just have chemical entities; we have all of these different types of entities. We've got data on nuclear test sites, neurons, stars. What do I see one in here? Fat types, right? So I'm guessing that is a lot of food related entity types. Just grabbing one at random, you can see fictional characters. We have what types of properties do we have for fictional characters? We have things you might expect: alternate name, what kind of appearances they've had in various animations, right? So we can take that fictional character type and ask for a random entity, and it'll give us Emrys Killebrew, who I don't really know for sure, but I can ask what do we know about it, and we can ask for its data set, and we can find out this is basically all the information that we have. I most certainly would have picked one. If I hadn't just called random entity, then I certainly would have picked one that seemed a little bit more interesting than Dr. Killebrew. But maybe that's not his fault, right? Maybe Octavia is a little bit more interesting. Well, so it's what I like to call non-rectangular data, right? So we've got a lot of different properties, a lot of different entities. Not every property is filled for every entity, but that's the basic idea. We could ask entity list at these fictional characters and see how many we have, but we're not going to spit them all out here in the notebook. I'm just going to ask how many, right? And that's just going to come back. We've got 11,000. There probably are some that are not in that backlit. I don't know. I've read a couple of fiction books, so. Right. So now we get back into the bread and butter, the meat of the talk here, the chemical entities, right? With chemical entities, I'm mainly interested in chemicals, elements, and isotopes, right? So I can sit here and I can ask how many isotopes do we have? And it's going to come out to be about 3,500, I believe. And I know that because we most recently scraped all the data, right? So random entity, the isotope, you can see that. And again, we're going to have its half-life, its atomic mass, the binding energy. And this data is regularly checked against, what do you call it, definitive sources. So there are specific places where we go to get the information about the half-life, the atomic mass, and everything for the isotopes, and similarly for chemicals and for elements. So you've got that list of entities. How do you end up finding the one that you need, right? So I could call random entity. I could look at all the entities. For elements and isotopes, it's very easy. It's calcium 56. But for a given chemical, you might not know what the standard name is. So you would do control equals. You do control equals here in a notebook, and you just type water. Let's see. I'm going to do a live demonstration. I made these into bitmaps, but here I'll just do control equals, and I'm going to say benzene, right? Bam, and that is benzene, and I can ask again for its data set. But I could also, look at it using built-in functions that are meant to work with molecular type entities. Right. So that's how you discover property or discover an entity. you can ask for its properties, right? So again, so there you could use the function, you could use a property to access the stored information in there. You can take those properties and then pass them into various functions in the language. Yeah, so we've got entity value, right? I'm not going to go too far into depth, but you can go through this notebook. You can find the sucrose, you can find all the elements that are in it, right? You can find all the known isotopes for those elements. It's a really good, it's a way to make things discoverable, right? Then we've got various properties, right? So for the elements, you've got the adiabatic index, the critical temperature, you've got various chemical and isotope properties. Although really, you don't need to remember these. You don't need to pull up the list and copy the yellow box, because you can just use, again, control equals. So we ask atomic mass of tritium, and it comes up, it parses the fact that you wanted the tritium, this isotope element or isotope entity, and then an isotope property. And then it comes back with the exact mass, right? I could do, benzene atom count. Yep. And again, it's going to come back and it'll evaluate, and it'll come back and it tells you it has 12 atoms, right? For various types of entities, there's usually some kind of image property, right? And these are the kind of properties that are going to be consistent across the various entity type domains. So that's how to interact with chemical entity types. Now, just to step back a little bit, something that grew out of the chemical entity types was this built-in chemical objects into the Wolfram language. So it started back in version 12, where we have all these new data types to represent chemical species, right? You can have an arbitrary atom, a bond. Those go in to make up a molecule. You can have a molecule pattern for various functional groups, like a carbonyl molecule pattern. And then starting more in version 13, we started to have support for biosequence, where you can have arbitrary, you can represent an arbitrary amino acid sequence or an arbitrary DNA sequence, and you can find its properties. We started to look at chemical reactions. We introduced a symbol for like a type of reaction, a reaction mechanism almost, so a pattern reaction, which takes in molecules and spits back whatever the product would be. We introduced a data type to just represent a chemical formula. So all you know is the chemical formula. You can still do quite a bit of computation on that. And then more recently, we introduced the chemical instance, where you can say 5 grams of HCl or 2 moles of nitrogen at 77 Kelvin. And so here's the way it works, basically. I mean, the most simple biosequence, this is kind of a silly example. This one just has the one letter. You can do a peptide sequence with just the one letter T, right? Which stands for L-threonine. So you say biosequence, and you get the object back. You can convert that into a molecule. Because it was just one peptide residue, then you can find that entity, and it is in the knowledge base. It's under L-threonine, and you'll find in the knowledge base lots of lots of curated properties, like experimental properties, like the melting point, the density. I'm not actually sure if we have the density of L-threonine. Yeah, we do. But that's not something that you could compute from the molecule object, right? Because the information isn't there. It requires experimental work, right? So density, it's not an available molecule property. So you can take that guy, you can make him into a chemical formula. And again, we're not gonna know the density just from the chemical formula. But we can say the element mass fraction. And we can find out just based on the chemical formula that this guy is 40% carbon, 7% hydrogen, 11% nitrogen, and 40% oxygen. And then finally, we can look at these. We can say, does that biosequence match this chemical formula? Yes. So that is reasonable. So that those are the parts that were put into the language between version 12 and 13.3. And since then, we've also been working on Wolfram chemistry packlets. So the Packlet repository is a new, long-requested feature for the language. It's where we host all these various external and internal packages that aren't part of the system context, right? Fast turnarounds. We can bring functions geared towards education or niche functions that wouldn't make it into the system. And so in chemistry realm, we've brought a Packlet for computing fingerprints of molecules, for generating isomers. This one is actually pretty neat. I'm not going to talk about it at all. Self-referencing embedded strings, and then protein visualization, and then just various chemistry functions. So with that, I'm going to dive in and just show off a few of these interesting ones. So a molecule fingerprint, it's a numerical representation of the features in a molecule. When you encode those features to a sequence of bits, they allow fast and meaningful similarity comparisons. The functions that are used by the molecule fingerprints Packlet are the same ones used by feature extract. So when you do a feature space plot on a list of molecules, it'll be doing the same, using the same functions that are exposed in this Packlet. So for example, how do you determine how similar two molecules are, right? So we have these two molecules. Okay, I mean, I could see this one has a carbonyl group. This one has some carbonyl groups. They both got some rings. They might have these two bridged six-membered rings, or fused, I think is the right term there. But exactly how similar are they? Well, what we can do is we can do a fingerprint. We can check all the different functional groups that are available in those molecules, and we can plot them, right? So here we see that they both have lots of rings. This one has H-bond donors, whereas this one doesn't. This one has a dialkyl ether, and this one doesn't. So we can bring that in, and we can make a substructure key fingerprint of that molecule, and it says it has three members of this pattern, one member of this pattern, one occurrence of this pattern, etc. We do the same thing on both molecules, and then we find their Jacquard dissimilarity between them. And so the Jacquard dissimilarity is 0.71. So it means that these guys are pretty dissimilar. They would have a similarity of about 0.28. And so that's a way of quantifying the idea that we had when we looked at these molecules, that they're not all that similar. All right, I'm going to skip over hash-based fingerprints for this talk, but it's in the notebook if you find that interesting. And then we have isomer generation. I think this one is pretty neat, to be honest, right? So starting from a chemical formula, generate all possible molecular structures with correct valence. So if you start with C16H28, you wouldn't get any isomers that have 5 bonds to carbon or 2 bonds to a hydrogen, so it keeps all that stuff into account. And it just exhaustively looks at all the chemical graphs with those many vertices, and then assigns the correct atom type. And so with this Packlet, you could, in reasonable short amount of time, in 50 seconds, it came back and told me that there are 31 million different possible chemicals with the formula C16H28. Now, I did not try and enumerate them. I looked for a smaller formula, C8H12N2, of which there are only 2 million. And you can list them in just under a second. And then you could choose some of them randomly and plot them. And some of these are going to be reasonable chemicals, some of them not quite as reasonable. But the functionality of the Packlet is to enumerate all possible chemicals with a given formula. We've recently put out this Packlet by Sutik Saha, who works in the chemistry group here, for plotting 3D visualizations of proteins. And so here we do like to use the protein structure 6RB9 as a test case, because it's really quite beautiful, at least when you use the cartoon ribbon diagram. You can do a Ramachandran plot to look at all the dihedral angles in the protein, and all of these functions are available in the protein visualization Packlet. And to install it, you just have to do Packlet install and then needs to get it onto your context path, and then you have it available. And then finally, I'd talk about the Packlet that we just call chemistry functions. So these are interesting functions, useful in chemistry. Some of them are useful for education, some for research, some are just kind of neat, but they don't belong in the system context. And so that's why we came up with the chemistry functions Packlet. This one I actually just wrote in the past week, and then we published it right away to the Packlet repository. So alkane name. What do you call a straight chain alkane with 5,267 carbon atoms? Well, of course, you call it hepta, hexaconta, dicta, pentaliene. Right, of course. And you can then take that same string and convert it back to a molecule, because all of these different little pieces here are parsable, and we can read that and see that has the given number of carbon atoms and hydrogen atoms. We could ask for its molecular mass or how many element counts it has. Yep. You can create a random alkane with 267 carbon atoms and then plot it. And then you could find the systematic chemical name. And so I thought this was pretty fun to write, actually. That given a chemical like this, you can systematically compute the name to be this nice string here, and then using another resource function in our chemistry toolkit, you can convert that into a SMILE string. Let's see. Empirical formula. This is something that is covered in most high school general chemistry classes. So you're given a problem like a dye has a percent composition of 75% carbon, 17% nitrogen, and 6% hydrogen. Determine the molecular formula for the dye. So first you can get the empirical formula, and then you bring in the fact that it's 240 grams per mole, and you find out that it's in fact C15N3H15. This is something that definitely comes up in most any Gen Chem classroom. Smiles plot, I think, is pretty fun. So these, I only showed one briefly. Let's see, in the previous slide here. This is a SMILE string that encodes the molecular structure, right? So what it means is when you go down this is a carbon atom, carbon atom, carbon, carbon, and then you hit this parentheses, and that's going to be a side group, right? So the main chain here goes to here, right? So this is the main chain, and then side chains are all grouped inside parentheses. And it works for rings as well. You can really, using a function like this, inside your notebook, you can interactively explore what is meant by all these little notations inside a SMILE string. And so for students new to chemistry, this is quite useful. We've got functions for generating, for manipulating the 3D structure of a molecule interactively. So we have coordinates for this, but I got to tell you, I think that bond should be a lot bigger. Yeah, obviously that's not physical, but that's a pretty neat example there. Right, and then we've got also a function to draw molecules directly in your notebook. There we are. Yep. And yeah, that was me flying through all of the slides that I had prepared.
Yes. Yeah, somebody was asking about plotting isotopes in terms of spectra. And I responded they might be interested in the atomic line entity type, which has an example showing how to plot a spectrum of a particular thing. So,
thanks, Jeff.
Hello, this section will cover new biology data collections accessible in the Wolfram language. In version 14, new hierarchical entity type, taxonomic species, is introduced to describe biological organisms. Taxonomic species data is based on NCBI taxonomy database and provides scientific names, hierarchical relation information of near 2 million taxa. Taxa includes animals and plants, as well as bacteria, virus, and other latest classified microorganisms. In addition to taxonomical knowledge, curated collections encompass biological, geographical, and ecological information to characterize organisms. Latest Wolfram language functions allow advanced natural language lookups and analysis of diverse organisms. The taxonomic species entity type is hierarchically organized. Just like the tree of life, this tree diagram illustrates how the entity types are hierarchically structured. You can see that child types like virus and bacteria are placed below taxonomic species. In the Wolfram language, you can access the child types of a given entity type using this newly introduced entity type concept. So here you can see the list of direct children of taxonomic species entity type. And the taxonomic species type includes taxonomic information as well as common properties that are shared across all species domains. So here the list, and you can see the conservation status, nucleotide sequence links, and observation locations. Those kind of properties are included in taxonomic species entity. Child entity type inherit properties of their parents, but also include some unique set of properties that are specific to child entity type. In this case, I show the animal entity type. So here, for example, you can find the number of properties belong to each hierarchical entity type. So there are about 100 properties belong to taxonomic species, and about 70 properties are included in animal entity type. And you can also find the properties that are unique to the child entity types. So here, for example, properties are unique to the animal entity type, including reproductive properties, body temperature, and properties to describe behavioral characteristics. So those are listed here. Taxonomic species data provides more detailed taxonomic sequence information, including not only the primary taxa, but also intra-ranked level taxa. Like here, subphylum, superclass, suborder, so that level of information is available. So that allows us to visualize more detailed taxonomic trees. This taxonomy graph resource function is very useful to visualize the taxonomic tree. You can just plug in your species entity of your interest and get the nice here layered graph with tooltips. And this function takes all the options from the graph function, so you can also customize the style of the graph as you like. You can also compute a list of species that belong to a given taxonomic branch by accessing the species list property. So for example, there are 12 known species belong to elephants family, and here the list of those species. This analysis can be done for any taxonomic branch, so you can easily find the list of species that belong to the higher taxonomic level group. Finding a common ancestor for a given list of species is also important for taxonomic analysis. This taxonomy common ancestor function comes in handy for that. So here you can find the common ancestor for dragonflies and beetles. Other information you can access includes the dates of taxonomic classification. The dataset provides the original year of publication as well as the latest year of publication, because sometimes a given species can be reclassified and merged into another taxon or placed into a different branch in the taxonomic tree. And one of the key features in this new dataset is that you can conveniently analyze information about a folk taxonomy group. Fish, otters, monkeys, and so forth are so-called paraphyletic taxonomy groups. Like shown in this diagram, for example, fish species belong to multiple branches in the taxonomic tree, and there is no single taxon to describe fish, and therefore it is a folk taxonomy group. In the Wolfram language, fish is now represented as a new species phyletic group entity. And using this new concept, you can easily get a list of fish species by using this implicitly defined entity class syntax. This syntax allows you to aggregate values for the group. So for example, here you can find the weight of various monkey species and find their distribution. So this is the weights of various monkey species, and using the box whisker chart, you can actually see that monkeys' weights vary greatly by species, ranging from about below 1 kilogram to 24 kilogram, while median weight is 5 kilogram. So this implicitly defined entity syntax is very useful to classify organisms. Another key feature to explore is the geographic property of species. New syntax allows you to look up animals by observation locations. Any of the geo entities are supported for these lookups. So those include countries, oceans, as well as park, mountain, lake, volcano, and so on. So for instance, here you can find a squirrel species specifically observed in Zion National Park, and you can get the data set including their images. For ocean animals like dugong here, you can find the bodies of water where a specified organism is observed. And using geographics function, they can be visualized on a map as well, like this. Species can be ecologically analyzed, and this example here compares biomass across all species domains, including animals, fungi, archaea, and so on. So here is the bar chart you can see that plants and surprisingly bacteria share a large part of biomass, while animals cover a really small fraction of the entire biomass. Conservation status information is also available. This query explores endangered mammal species. And here are some examples. And then you can find the observation countries and the color countries by the number of endangered animals found. So here is the map used geo-region value plot. Biomes or habitats of organisms are also accessible as entities, and polygons are also available for all terrestrial biomes. So this map is illustrating terrestrial biomes, including desert, forest, mountain biomes, and those are colored here. Then going back to the endangered mammals, you can actually analyze their habitats. So this is coloring the number of terrestrial biomes for endangered mammals. And by comparing these two maps, you can see that more endangered mammals species are reported in forest, rainforest, and savanna biomes. Food habits are also an important aspect of animal characterization. Animals' food habits, specific diet, predator-prey information is available to explore. So here, for example, you can find animals specifically observed in Illinois that Cooper's hog prey on. And this the image collage of those, and you can see the small birds as well as rabbits, squirrels, and some reptiles are found. And making a food chain is also very easy. You can create a food chain of animals in a selected environment. In this case, animals specifically observed in Grand Canyon National Park are selected. And this layered graph plot illustrates the food network of those animals observed in Grand Canyon National Park. And you can see coyotes to chipmunks, squirrels to small birds. Those are illustrated in here. Other than food habits, other ethological characteristics like daily activity patterns, dormancy types, social behaviors, and so on also can be looked up as well. And you can also get a nice data set like this. So for example here, you can look up animals by their anti-predator strategies. And snake species that exhibit cryptic anti-predator adaptation are specifically retrieved here. And here's some example. And the image collage of those illustrated in here. Similarly, you can also look up animals by perception channels. For example, we are selecting fish species that can perceive a magnetic signal. Here are some examples and illustrated here. Also, it's noteworthy that the sound that animals make can be explored using this animal sound resource function. So here's just some example of bird vocalization. So vocal sound of birds, frogs, or noises of insects can be discovered. And here is just one interesting example showcased here using the machine learning tools function to visualize the features extracted from the selected bird vocalization. So I selected these 15 birds and plot them using this feature space plot, including hawks, ospreys, some owls, hummingbirds, and so forth. There are also other important biological properties of organisms. For example, there are very unique features of organisms, like here, some reptiles whose sex are determined by the environmental temperatures that they experience during development. So those are selectively shown here. Genetic aspect of organisms can be analyzed as well. Here you can find the top 5 organisms with the largest number of chromosomes. In this data set, you can find that ferns surprisingly have the largest number of chromosomes. On this next example, we're plotting the 100 organisms that have the longest nucleotide sequence. And here's the plot. If you are interested in analyzing genomic information of organisms, this reference sequence assembly acquisition number comes in handy. And this function returns the external identifier object that links to the NCBI website, where you can retrieve and download genome sequence for further sequence analysis. There is also a new resource function that is available to get the detailed genome report for a specified species. So here you can find the genome assembly information and details, as well as like total number of currently known genes, protein coding genes for this specified species, lions. Beyond animals, features specific to plants, fungi, and other microorganisms can be explored as well. You can find plants based on growth conditions and physical characteristics. This one is showing the list of annual white flowering plants growing in USDA hardiness zone 6 and grows in coarse soil. And here you can get the list of plants that you can grow in this growth condition. Information about the hardiness zone is also available. You can access map images like this, and also polygons are available. And if you're not familiar with the current hardiness zone in your location, you can also look up the hardiness zone by zip code as well. For fungus species, there are mushroom-specific characteristics like spore print color, physical characteristics like stipe type and attachment type that can be explored as well. And this example is specifically showing psychoactive mushrooms that have white sprint colors, spore print color. There are microorganisms that infect humans, animals, as well as other plants. Here, for example, you can explore microorganisms that specifically infect various crops. There are about 5,000 species reported, and here are some examples. Then you can compare phyla of organisms that infect crops. And this bar chart is illustrating that many fungus species, as well as some bacteria, some virus species are known to infect crops. And of course, there are other microorganisms that are directly associated with humans. These are human gut microbiota that you can explore. And this data is specifically showing their associated body sites, where they are found in the body, and also sorted by their nucleotide sequence lengths. So there are a lot of interesting analysis you can make with this data set. So just a brief summary, new species data collections encompass knowledge of various organisms and hierarchical entity type syntax, and improved data lookup support in-depth biological data exploration in Wolfram Language. I also listed some links here that you can find more information about the new biology-related resource functions. Thank you.
Okay, so the summary of the talk today is the following. So we will first review the functionality for Astro Computation recently introduced about a year ago in Wolfram Language version 13.2. Fundamentally two functions, astroposition and astrographics. Then we will spend most of the talk discussing the update of solar eclipse, essentially a rewrite, which is a function that allows you to compute lots of things about solar eclipses. We will discuss a little bit how it works internally through a technique called Besselian elements, and we will conclude discussing or reviewing what we have discussed and talking about future work. So, as I said, we have recently introduced Astro Computation functionality, and one of the fundamental things that you want to do in Astro Computation is to find out the position of celestial objects. So, for example, this is the position of the sun currently given in horizontal coordinates, so azimuth and altitude. Here you have more information like the current distance to us, so our particular location on Earth, which is this one. This is the coordinates that we are using, and this is the date of this value. So astroposition knows lots of other things, and for example, it can take relativistic corrections into account, it can use other coordinates, many other things. You can also call it in real time, for example, updating every 0.1 seconds, and here you see how the sun is moving in the sky. And this is mostly due to the rotation of the Earth. We can compute also the equatorial coordinates of the moon, for example. Again, here we have the distance, the very precise frame that we are using, etc. Notice also how we use entities. So we access the Wolfram Alpha knowledge base that we have seen before, and Jeff Bryant, who is here today with us, has collected lots of families of celestial objects, moons, stars, planets, galaxies, etc. Here we can also see the real-time motion of the moon, and now this frame is not rotating, so we see here the actual motion in the sky. We can also construct maps of the sky. So this is the map of the sky, 25 degrees around the star Betelgeuse. And so we see constellations, we see other stars. We have some lines. This is the projection of the equator of the Earth, so the celestial equator. In yellow, we have the ecliptic line, so the path of the sun through the sky. We can add illustrations. We had an artist producing all these illustrations for the 88 constellations for us. Or we can, for example, select a different area of the sky and show a background image, what we call the galactic sky. So for example, here we are selecting a point in galactic coordinates. This is another of the coordinate systems or frames that we can use together with equatorial, horizon, ecliptic, etc. We draw a white point, large, which is here, at the coordinate 0, 0. There is also a frame of grid, of line, which is, of course, associated to the galactic frame. So there are some lines here. And so I don't know whether it goes to the screen, but here you see 5 of these squares, each of 5 degrees. So completing the 25 degrees circle that we are covering with this image. Okay. You have access to all projections in the Astro projection engine as well, like 150 projections or so. And what can we do for solar eclipses with the functionality available since version 13.2? So solar eclipse, which is a function we will see a lot more in the rest of the talk today, gives us the date of maximum eclipse for the next eclipse, which is going to be a total solar eclipse on April 8th. So we take that date, we observe from here, we use equatorial coordinates, and we ask geographics to show a map of the sky of only 1 degree of range for that date. And then what we do here is that we manipulate these from 1 hour before the maximum eclipse, 3 hours later. And so what we see is the moon going, and because we observe from Champagne and there is no totality in Champagne, there is a little bit of the sun that will not be covered. If we went, for example, to Indianapolis, we would see the sun fully covered. Right? So this is very precise computation. Or we can ask geographics to do the same thing, but now we observe from the sun. We are using ecliptic coordinates. So we see here the path of the Earth in yellow. We observe from the sun. We do the same trick of moving a few hours. And now we have to restrict to a very, very small range because from the sun, the Earth is very, very tiny. We do the same thing. And here we see how from the center of the sun, we will be able to see the moon passing in front of the Earth. Okay. Now, another thing that we have added in version 14 is much better access to timing of the moon. This is very important to control eclipses. So for example, we have a new function called new moon. It gives us the exact moment at which the moon will have the same ecliptic latitude of the sun. And so at that exact moment, which is very well defined, we will have new moon, which is on February 9th. Now, I wanted to mention that some calendars, the lunisolar calendars or the lunar calendars, are following the motion of the moon, such that months correspond to new moon to new moon. And for example, the Jewish calendar is one of those, and so new moons always fall near the end or the very beginning of a month. So in this case, it will be day 30 of this month. Or the Chinese calendar. And the Chinese calendar also, day 30, this means last month of the year. And we know that on the 10th of February, they will celebrate the Chinese New Year. We can also compute the dates of new moon, of course. We have a beta function, which is called lunation number. This is a continuous count of lunations, meaning new moon to new moon, counting 0 as the first new moon of year 2000. We can see this counter advancing in real time, and this means one here is a full round of the moon around the Earth. Right? Okay, so why are new moons important for us? It's because every solar eclipse happens close to a new moon. It's not true that every new moon has an eclipse, and that's because sometimes the moon passes, like, for example, on January 11th, the moon was here. This is about 5 degrees. And you see, if you evolve a little bit of time, you see how the moon was passing about 5 degrees from the sun. But sometimes it happens, and this will happen on April 8th, the moon does pass just in front of the sun. And depending on where you are on Earth, this exactly covers the Earth. Okay, so let's go now to the function solar eclipse, which, as I said, has been essentially fully rewritten. So we have extended the coverage a lot. We now extend the coverage to everything that NASA provides, which is this range of minus 13,000 to 17,000 years, so about more than 30,000 years. We have analyzed all solar eclipses. There are from 2 to 5 every year. So in total, there are more than 72,000 eclipses. And so this function gives you access or computes in real time every property, and there are about 100 properties about each of these solar eclipses. Properties involve things like dates, position, curves, polygons, types, etc. And before we go on, I'm going to introduce a bit of terminology. So here we have the sun. That's the moon going around the Earth, passing in front of the Earth. The shadow is projected on the surface of the Earth. And you see how the shadow has several parts. There is the umbra, this part. The parts of the umbra hitting the Earth will see totality. There is penumbra, this part here. The parts of the Earth seeing penumbra will see partiality. And then there is this part called antumbra, which is kind of the other side of umbra. It's not relevant for computations, but when it happens that the moon is a little bit farther away and it's antumbra what hits the Earth, then you see an annular eclipse. Something very important is to notice that this diagram is not to scale. It's really, really not to scale. So if that's the sun, and this is in millions of kilometers, that little circle there is the orbit of the moon. The sun, the Earth is at the middle. We enlarge that little circle 400 times, and now we see the moon there and the Earth there. So that curve there is that little circle. So you see how extremely rare is the fact that this shadow touches the Earth, right? Because this is so tiny and this is so far away. So with solar eclipse, we can compute global properties, like, for example, when is the maximum eclipse, which type it is. For example, we can find eclipses of various types: total, annular, hybrid, partial. And you can say from when. For example, this was the previous eclipse to the first day of 1900. So this means look backward in time. And so this gives you access to these 72,000 eclipses that we were discussing before. We can compute maps. And so we call geographics, and we put here various primitives. So for example, that's the path of totality for the eclipse on April the 8th. This is the area in which you will see partiality. Here you have the percentage of partiality. This is where the eclipse starts. This is where the eclipse ends. And there are various points that are very important for computation, the contact points. So we will discuss them a little bit later. There is always an instant of maximum eclipse, which is where the moon is closest to the shadow of the moon is closest to the center of the Earth. That's precisely what we report by default. And this is the position of totality, of maximum eclipse. So now, what happens during an eclipse? So this little circle there is the umbra. This is here is the penumbra. And then what happens during an eclipse is that the shadow approaches the Earth. It touches first there in that red point. Then it moves. And you see how the shadow is moving linearly, but when combined with the rotation of the Earth, the path at the end has this shape. So that point there is maximum eclipse, which for this eclipse is going to happen in Mexico. And then that's where umbra touches Earth for the last time. And this is where the penumbra touches Earth for the last time. So these are called P1, P2, P3, P4. Okay? So it's important to have this in mind for visualizing what happens with an eclipse. So we can have eclipses of various types. For example, this is an eclipse of annular type. And this is an eclipse in which the Earth is not going to be completely, the shadow of the moon is not going to cover all of Earth or be inside rather Earth, right? That part there will not touch Earth. And that's why these parts now are together. There is some continuous line there. This is an annular eclipse. And at the moment of best eclipse, of maximum eclipse, you see how the moon will look smaller than the sun, right? Because the moon is in an ecliptic orbit. Sometimes it looks smaller than the sun. Sometimes it looks larger than the sun. Something very interesting for eclipses is that there are cycles. Because this is all a result of a geometric configuration. After a number of years, the geometric configuration more or less repeats. For example, there is something called the Saros cycle, already known to the Greeks like 2,000 years ago or more, very impressively. And these Saros cycles are numbered. So the eclipse that we are going to see in the US in April belongs to Saros 139. If we put all the 71 eclipses of this Saros cycle together, we see that they range from 1500 to 2750, so more than 1,000 years. We compute the differences between the dates, we compute around, and then we see that very, very precisely, they are all separated by one Saros. And one Saros is defined as that exactly number of dates. So this is 22.3 exact revolutions of the Earth, of the moon around the Earth. And it's exactly or nearly 242 what's called draconic months, which is related to how the plane of the motion of the moon is rotating, is moving with respect to the plane of motion of the Earth, the ecliptic. So this eclipse we will see is number 30 in the Saros cycle. And something very interesting is that we can put together the magnitudes, so how much the sun is covered by the moon, and you see that the first eclipses of the Saros do not cover, are partial. Then we get total and annular eclipses, right? This is one, so full cover, and then this is, and this is a very interesting plot in which we just put the various eclipses and where is the path of totality. And we see how it looks very similar for all these members of the Saros. Right? So that's a family of eclipses. We can now, again, take that eclipse. We compute another cycle, which is called INEX. And now we are in index 55. There is a property, Saros index, that gives you simultaneously the Saros number and the index number. Now we do this for all eclipses between year 1000 and 3000. And we have this number of eclipses. Now we put them all together in these graphics. Each of these dots is a point. And or horizontally, we have the Saros cycle. So each of these are Saros cycles, Saros families. And here in horizontal lines, we have index families. And that's our eclipse, which is there in the middle. Right? So if you take any eclipse and you move vertically a little bit, that's about 18 years. If you take that eclipse and you move horizontally, that's about 29 years, an index. It's very interesting that they can all be classified with these two numbers. Local circumstances is the name that's given to compute things from where you are. And so, for example, now we're going to see when is this eclipse reach its maximum from my location here in Champagne. And it's that date. So this is the best moment. But we saw before that the local magnitude, the percentage of coverage, is not going to be 100%. It's only going to be 98%, which means from here, from Champagne, we still need glasses to see the eclipse. You really have to be in totality to see the eclipse without glasses. You can compute the contact times. So the first time that that we will see the moon touching the Earth, etc. The sun. And here we have a curve of magnitude. So that's the center point. That's this instant here. And an hour and 20 minutes or so before the moon will start covering the sun. That's where it will reach the maximum, about 98%, and then it will decrease. And for example, you can also compute what is the shadow of the moon at a given time. So I'm going to do that for these 3 dates, for these 3 moments, right? We get these ellipses, right? Because of the perspective of how the surface of the Earth is placed with respect to the moon, it looks typically elliptical. And so we just call geographics, and we get those 3 moments, right? They are separated by 1 minute, right? So you see how fast the shadow of the moon is moving minute by minute with respect to where we are here in Urbana-Champaign. And then to finalize, I want to repeat how it's all computed. We use a technique called the Besselian elements. It's a technique introduced by Friedrich Bessel in 1824. So actually, in a couple of months, we could celebrate the 200th anniversary of the Besselian elements. And interestingly, it's such a good technique that it's still used today to compute all these things. So what it does is taking the Earth. I mean, the Earth is nearly spherical, so I'm just exaggerating here the ellipticity of the Earth. And this simply constructs a frame which is adapted to the shadow of the moon. This is the shadow. Right? And so just by being very careful at the construction of this frame, you make all these computations a lot easier. So this frame moves. If this is the moon moving in front of the Earth, we can think that this is the Besselian frame. And so as the moon, you see how the frame is rotating. It's always been adapted to having this plane perpendicular to the shadow of the moon, to the axis of the shadow of the moon. So if you want to compute anything about an eclipse, like again, our eclipse here, you just call the Besselian elements functions. So solar eclipse will compute them for you. This is just using Astro position computations internally. And then once you have these functions here, these are functions of hours from a given point, this point here, then you can compute anything. Like, for example, when is the distance minimum from the center of the Earth and the shadow axis? We compute that, which is this distance, just an Euclidean distance. And now the only thing we have to do is to find the minimum of that distance. We find the minimum, we convert it into a date, and we get that date. Now, if we convert our result before into the TT frame time system, the terrestrial time system, this is just a matter of being very precise about what we mean by time, we get exactly the same result. So again, any computation that might be needed for an eclipse can be computed from these functions here. This was just a very small example. Okay, and to conclude, I'm going to mention just that Wolfram Language 14 has extended the astronomy computational power of Wolfram Language. We started adding functions for Astro computations in version 13.2, and now in each version, we will add something else. So in this case, it was solar eclipses. So it has been, this function has been fully rewritten. Now we handle more than 72,000 eclipses from these years. And these are the years in the coverage of the most precise ephemeris data that NASA provides, which is what we use for Astro computations. There are lots of properties for them. We are now updating our precisioneclipse.com site that you will be able to use to compute all properties and nice maps about the eclipse of April the 8th. And talking about eclipses, I wanted to mention that Stephen Wolfram has just written a book about solar eclipses. Here we have a presentation in our community, and here I have a copy. And I truly recommend to read this book. It's a small book that has a first part with qualitative explanations about eclipses, etc. And then it has 3 appendices with lots of computations, maps, and very interesting data about eclipses, about this eclipse and others. And it's all computed with the function solar eclipse. And I think that's all from me. Thank you very much. Can we displace sky maps according to distance to objects in 3D? Well, Jastrographics takes into account distances in order to know what to put in front of each other. And then UJEF has a very interesting function in the Wolfram function repo to show images in 3D. And do you think that's what's meant here?
I don't think so, but I'll give me just a second here, and I'll put a link to that, the one that you're talking about. Astrographics is basically 2D only. It's essentially it's projection. So, sorry, I'm doing 2 things at once here. Let me just copy the. So,
can you generate a sky map with a magnitude limit of or range? Magnitude. I don't understand this question. Magnitude limit.
So they want to.
I see.
Yeah.
Yes, magnitudes in stars. Yes, we can change. There is a parameter inside the in the code of Astrographics that allows you to have more stars. We have a collection of up to 100,000 stars, and Astrographics typically tries to show at most 1,000 stars. But yes, it's possible to increase the magnitude and so the 100,000 stars in sky.
I put a link to my telescope view Wolfram function repository item in the chat if anybody's interested in that. If you're interested in like looking at a planet, like how it would look from Earth or from some other body, you can even look from like a crater on Io if you wanted to. But the point is you can say, what would it look like? And it is done in 3D, but it just gives you a 3D view of solar system objects for the most part. So Jose's showing it on his screen there. So we have that ability to do that. And then, like I said before, we have astrographics, which is mainly 2D. We don't have anything specifically called, what was the terminology that they used? Anaglyph, side-by-side images. I'm not quite sure I understand what's being asked for, but those are the 2 things we have, is astrographics and the telescope view 3D. Keep in mind, this is all very new. Starting in 13.2 is when astrographics was just introduced, and it's still experimental. So we're hoping to add to that in the future, but obviously more feedback we get as to what kind of features users want, and that would be useful.
And then there is another question. Do we know when the book will come out on Kindle? This, I don't know. We would have to check with our publishers. But I know I can show that it's already in paper, and it looks very nice.
I just checked with Paige about the Kindle version, and she says yes, that is coming very soon, perhaps in the next week. So the next week or so, if you want to check back on that, there very well should be a Kindle version available.
Great.
Somebody asking, can it be used for graduate astrophysics? I'm not sure what it is. Obviously, you can use Mathematica for graduate level research.
Yes, we have lots of functionality, and there are other functions we haven't shown to compute astro distances,
angular
separations, and other properties. And as I said, this all takes into account various effects, like the fact that we are on the surface of the Earth, so you can see the effect that has on angles as we rotate with the Earth, relativistic corrections, so the fact that light propagates at the speed of light, it's also taken into account. Even the gravitational deflection by the sun.
Right. Like Jose touched on this earlier, but astrographics, you're not just observing from Earth, but you can, with different astro frames, you can actually observe from the solar system barycenter or from various other places. So you have to keep that in mind when you call for astrographics. It's quite flexible. I don't know much about Apple Vision Pro. I've heard of it. It's kind of a, I think it's going to be the new new iPad or iPhone, I guess, that's going to be coming out of the new hot Apple item. I think Stephen Wolfram actually recently ordered one. I don't think they're very new. I haven't ever used one, but I think we're probably going to play with them in-house to see what we can do with them. But I don't think we have one in-house yet. I'm not sure if they're even officially out yet, or if there are, there's probably a backlog on orders of them.
I'm hoping that Wolfram Language itself will have some sort of link or connection to it, and then when that happens, we will certainly use it for us.
Somebody says they already have one, so I guess they are officially out. So